Hi, I'm here to talk about the gut microbiome in states of health and disease. And the organizers have asked me to specifically address the challenges and opportunities for developing microbiome-based diagnostics and interventions in the era of precision medicine. So I have no disclosures to make that are relevant to this talk. So let's start with the definition of a microbiome. A microbiome is the totality of microbes, their genetic elements, that is genomes, and environmental interactions in a defined environment. So it includes not only the microbes, but the ecosystems that they're in. And this is important to know because we have several microbiomes that are part of us. For example, we have a skin microbiome, there's a microbiome in the lungs, but far and away, the largest and most diverse microbiomes are in the gastrointestinal tract. And here you find microbiomes from mouth to anus, and each are very different because the GI tract is comprised of multiple ecosystems that are connected in series. Each of these microbiomes provide very specific and essential functions for us. So I view the gut microbiome as a microbial organ. Why? Well, we absolutely need our gut microbiome for health. We can transplant it. And when transplanted, it can even be rejected by the recipient. So in many ways, it is an organ system of our body. It's comprised of trillions of microbes. They're shown here in this figure on the left that live in close proximity to our own cells. And like other organ systems, the gut microbiome can exist in states of health as well as disease. In a healthy state, the gut microbiome provides many essential functions that are important for our well-being. Some are shown here. For example, in the modulation and regulation of our immune system, nervous system, and other organ functions, as well as biosynthesis of important compounds like vitamins, amino acids, and also in nutrient digestion and absorption. In a disease state, the microbiome can cause or contribute to many human disorders. For example, inflammatory bowel diseases, metabolic neurodegenerative disorders, atherosclerosis, and cancer. But what about HIV? Well, here I think the data of many studies is summarized very nicely in this table that came out of a recent review. And you can see there is a general pattern of microbial populations that go up or down that would be indicative of what we call a microbial dysbiosis. And this is across many populations and many types of studies. For example, we see this dysbiosis in HIV as well as the MSM populations. However, you can also appreciate that there are differences from one study to another. And that is probably accounted for by the fact that different controls, demographics, clinical situations like environment, diet, medication could lead to this variability. Ultimately, we have to ask ourselves, what do these changes mean? So let's look at the possibility that these changes are relevant to the development of clinical symptoms or disease. So there have been several drivers of HIV and MSM associated gut dysbiosis that have been identified that could potentially impact the host. For example, diet, sexual behavior, antiretroviral therapy have all been shown to have direct impact on the gut microbiome by either increasing pathobionts, these are commensal organisms that, given the right circumstance and opportunity, can transform to a disease-promoting state, or through the decrease of immune regulatory bacteria. Collectively, these actions can lead to dysfunction or breach of barrier function of the gut mucosa, allowing transmigration of microbes and their mediators that leads to a cascade of events that starts with innate immunity, followed by activation of adaptive immunity, which then can cause both subclinical or clinical inflammation, and through a feed-forward mechanism lead to worsening of the dysbiosis and possibly to the development of clinical symptoms and disease. So we talked about how these actions might cause or contribute to clinical symptoms and disease. But these changes could equally be accounted for by being an effect or consequence of the condition of the patient. We have to recognize that the gut microbiome is very sensitive to the conditions of its host and environment. Now, we can take advantage of that because in many ways, the gut microbiome is like the canary in the coal mine. In this case, the coal mine is the host. So what happens in the gut microbiome could reflect clinical states of the host. Supporting this notion are two studies that I'm going to talk about. One was published several years ago by the MGH group, where they looked at the structure of the gut microbiome and its metabolic activity in inflammatory bowel diseases. And the study involved non-IBD controls and IBD subjects with either Crohn's disease or ulcerative colitis. Here, multi-omic screening of stool samples was performed. Metagenomic shotgun sequencing was used to define microbial taxa, genes, and pathways, whereas LC mass spec was used to measure lipids, polar metabolites, free fatty acids, and bile acids. And you can see in the ROC curve on the right that whether you look at these parameters individually or in combination through either cross-validation or independent validation studies, you can see they all perform quite well. And what this would suggest is that these multi-omic parameters can define or even approximate the states of health. Now, arguably, one wonders whether this has clinical utility, considering that we have very good conventional diagnostic tools to determine who has IBD or who is healthy. But I think that this is the current state of the art, and with advances in technology, this may eventually reach a level of clinical application. Now, the same group published another paper more recently, where again they performed multi-omic studies to determine if there were microbial determinants associated with responses to biologic therapies in inflammatory bowel diseases. So in this study, they obtained blood and stool samples as baseline measures prior to treatment of patients with moderate to severe IBD with anti-cytokine or anti-integrin therapy. And then clinical outcomes were tracked to determine who were responders or non-responders. The measures that were used included stool metagenomics, serum metabolomics, and serum proteomics. And as you can see by the volcano plot on the left, they found a number of highly informative markers that could predict states of remission or non-remission. I think this is really a step in the right direction, showing the potential of these types of measures in predicting outcomes for patients undergoing specific therapies. It's still imperfect, though. And so the question is, well, how do we advance the field? I think that we really have to answer the question, what is a healthy microbiome? Now, this seems like it might be obvious, but so far, nobody has come up with a definition that works for everything. People have tried to define a healthy microbiome by taxonomy. They've used diversity measures as a metric. They've done studies or measures of stability, resilience, and connectiveness of the microbiome. And then they define it as whether a healthy microbiome is in a healthy patient. But then again, what is a healthy patient? And if you look at many metagenomic analyses of a general healthy population, you will see tremendous diversity that is very difficult to define as or use as defining healthy. The problem lies in the following. If you look at the figure at the right, if we're looking at these trillions of microbes and trying to define their genetics and their properties at a very individual level, this is a very daunting task because you get so much information. There's also so much variability genetically and functionally, even among microbial species. And there's tremendous inter-individual variation of the microbiome. But we know one thing, and that is that these trillions of microbes have something in common. They make a finite set of metabolites. And arguably, these metabolites and small molecules are more important because they directly impact our biology. For example, they're involved in the regulation of our immune functions and the functions of many organ systems. So metabolites can provide a very practical and measurable quantity that could assess the states of health or disease of the gut microbiome. And this has become the priority of my laboratory in defining sets that perform well in specific contexts and specific indications and diseases to be used as a diagnostic tool. Now, there are other approaches to consider when we're trying to evaluate the impact of the gut microbiota. Certainly, the best of them would be direct studies of human subjects. But for both technical and ethical reasons, our ability to go beyond association and correlation is very, very limited with human subjects research. So then we can use experimental models, for example, animal models. And here we can answer, or we can ask and answer specific questions. However, all of us recognize that humans are not mice, and we differ considerably in our physiology and responses. More recently, many of us have turned to organoids. These are cell lines that are derived from intestinal or other organ system stem cells, which grown under specific growth conditions can lead to the generation of sort of mini organs. And this technology has really advanced the field because these lines now become living archives of our patients. And they've been very useful for studying both pathophysiology, gene responses, responses to therapies, as well as gene editing that could be eventually used to correct genetic disorders through autologous transplantation. I want to end with these takeaway points. The first is that the gut microbiome plays an important role in health and disease. Second, it's important that we try to define what is healthy versus unhealthy. This remains a challenge, but I think it can be eventually resolved. Gut microbial membership and function are greatly affected by the clinical state of its host. We can take advantage of that because many predictive markers may emerge from the study of the gut microbiome that will be clinically useful. Disentangling cause from effect with regard to alterations of the gut microbiota remains a formidable challenge. I think we're going to eventually resolve this. Technology and informatics are constantly improving, and I think we're going to see the light at the end of the tunnel. Finally, multi-omic analysis of clinical samples showing promise for the identification of informative biomarkers and precision interventions. With that, I'd like to end. Thank the organizers for inviting me, and thank you for your attention.